Hello students, we'll move to the next session. In this session, we'll discuss the first reproductive event which takes place in sexual reproduction, that is gametogenesis. So you already know that gametogenesis, the formation of haploid gametes, that is haploid male and female gamete by the process called gametogenesis. There are two types of gametogenesis. One is spermatogenesis, the second one is oogenesis. The spermatogenesis process takes place in the male gonad or male primary sex organ, whereas the oogenesis process takes place in the female gonad that is ovary. So one contrasting difference between spermatogenesis and oogenesis is the spermatogenesis process starts after attaining puberty or sexual maturity, whereas oogenesis process starts during fetal life and completes after attaining sexual maturity. So let us start with the first one, that is spermatogenesis. So the question arises, what is spermatogenesis? The process of formation of haploid male gamete or sperm from diploid primordial male germ cells or immature spermatogonial cells that begins at puberty. The next question arises, when it does takes place. The process of spermatogenesis takes place within the seminiferous tubules of testes under the influence of two gonadotropins, one is FSH and LH. And you already know that FSH, which is commonly known as gametokinetic factor, because it initiates gametogenesis. For formation of gametes, we absolutely required of follicle stimulating hormone and luteinizing hormone. The hyposecretion or less secretion of these two hormones that ultimately affects to the process of gametogenesis. So the process of spermatogenesis completes in two steps. That is, first one is the formation of spermatids, and the second one is oogenesis, or spermiogenesis. The formation of spermatids includes three phases. One is a multiplication phase or proliferation phase. The second one is growth phase, and third one is maturation phase. The second step is spermiogenesis, the process of transformation of non-functional, non-motile spermatids into motile and functional sperms. So let us start with the first one, the formation of spermatids. So you already know that the seminiferous tubule is lined by germinal epithelium. In the germinal epithelium, there are two types of cells are differentiated. One is primordial germ cells, male primordial germ cells. They are also called spermatogonial cells. The second one is Sertoli cells. After attaining puberty, the primordial germ cells undergo mitotic division and meiotic division, produce haploid male gametes by the process called spermatogenesis. During the first step, the primordial germ cells undergo mitotic division and completes two meiotic division, and As a result of this, haploid spermatids are produced. So during the multiplication phase, after attaining the puberty, the primordial germ cells, under the influence of follicle stimulating hormone, it undergo repeated mitotic division. Repeated mitotic division. So as a result of repeated mitotic division, the large number of sperm mother cells are produced, which are diploid in nature. And this phase we call it as a multiplication phase. So soon after multiplication phase, the sperm mother cells accumulates accumulates energy nutrients and differentiates into primary spermatocyte. And this phase we call it as a growth phase. So after multiplication phase, after multiplication phase, the sperm mother cells accumulates energy nutrients and chromatin and become highly enlarged differentiated cells, and these enlarged differentiated cells are called a primary spermatocyte, and this phase we call it as a growth phase. So soon after this, the primary spermatocyte undergo two first meiotic division. So as a result of first meiotic division, the diploid primary spermatocyte is converted into haploid secondary spermatocyte. As a result of first meiotic division, the primary spermatocyte undergo first meiotic division. It produces two equal sized haploid secondary spermatocyte. Haploid equal sized secondary spermatocyte. And you remember one thing. So meiosis 1 is a reductional cell division. As a result of meiosis 1, diploid primary spermatocyte are converted into haploid secondary spermatocyte. Soon after the formation of secondary spermatocyte, these two secondary spermatocyte undergo second meiotic division, which is nothing but mitotic division. As a result of this, four haploid spermatids are produced, and this phase we call it as a maturation phase. And remember, at the end of maturation phase, it completes the first step. Okay, as a result of this, from single primordial germ cells, from four haploid non-motile non-functional spermatids are produced, and these non-motile non-functional spermatids are transformed into motile and functional sperms by the process called spermiogenesis. Spermiogenesis. Okay, so this is a process called spermiogenesis, where non-motile, non-functional spermatids are transformed into motile and functional sperms called uh, motile and functional sperms called spermiogenesis. So look at the primordial germ cells divides repeatedly by mitosis to form diploid spermatogonia or sperm mother cells. Each spermatogonia enlarges to form primary spermatocyte due to accumulation of nutrients and chromatin. So after growth phase, the primary spermatocyte undergo first meiotic division that results in formation of two equal sized equal secondary haploid secondary spermatocytes. And these secondary spermatocytes completes the second meiotic division that results in formation of four haploid spermatids. So these develops into immature spermatozoa, also known as sperm cells. Thus, the primary spermatocyte, a single primary spermatocyte gives rise to two secondary spermatocyte, and two secondary spermatocyte undergo second meiotic division that results in formation of four haploid spermatids. So until then, for development of these spermatocyte, they required continuous source of energy. That source of energy or nourishment is provided, or nutrition is provided by the nerve cells or sustentacular cells or Sertoli cells. That means the Sertoli cells provide nourishment to the developing sperms. So soon after formation of spermatids, the spermatids undergo spermiogenesis. Result of this, motile and functional sperms are produced. Soon after the formation of functional sperms, soon after the formation of functional sperms, the functional sperms are released from the seminiferous tubule by the process called spermiation. Okay, so this completes the process of spermatogenesis. Next, we'll move on to the hormonal control of spermatogenesis. So you already know that the spermatogenesis process is under the control of two pituitary hormones. One is FSH and the other one is LH. In turn, these two pituitary hormones are under the control of one hypothalamic hormone, that is two hypothalamic hormones. One is gonadotropic releasing hormone, the second one is gonadotropic inhibiting hormone. So after attaining puberty, the hypothalamus produces gonadotropic releasing hormone, that is GnRH, that stimulates the pituitary gland to release gonadotropins, that is FSH and ICSH, interstitial cell stimulating hormone, or luteinizing hormone. The FSH stimulates the primordial germ cells to produce sperms, whereas luteinizing hormone or ICSH, interstitial cell stimulating hormone, stimulates interstitial cells or Leydig cells to produce testosterone. And you remember, the testosterone is a male sex hormone or testicular hormone, which is absolutely required, or it is essential for the formation of sperms. One, and also responsible for growth and development of male reproductive system, and appearance of secondary sexual characters after attaining sexual maturity. Okay, so under the influence of FSH, the Sertoli cells develop androgen binding protein, and this androgen binding protein which concentrate testosterone within the seminiferous tubules. So excess of testosterone inhibits the luteinizing hormone production by the anterior lobe of pituitary, and GnRH production from the hypothalamus. That means when the sperm production is under control, immediately the Sertoli cells secrete inhibin hormone. That inhibin hormone inhibits FSH and LH secretion from the pituitary gland. So as a result of this, when the sperm production is under control, inhibin hormone stimulates the hypothalamus. So as a result of this, as a result of inhibin stimulation, hypothalamus releases gonadotropic inhibiting hormone. As a result of this, the secretion of FSH and LH temporarily stops. And the normal release of testosterone is under the control of negative feedback control or negative feedback mechanism. There are two types of mechanism. One is a positive feedback mechanism, other one is a negative feedback mechanism. So what is positive feedback mechanism and what is negative feedback mechanism. If response increases the original stimulus, then it is called negative feedback mechanism. If response increases, sorry, if response decreases the original stimulus, then it is called a negative feedback mechanism. If response increases the original stimulus, then it is called a positive feedback mechanism. So the production of sperm is under the control of okay, it is a negative feedback mechanism. Negative feedback mechanism. The this is about the hormonal control of spermatogenesis. Look at the picture. Hypothalamus releases GnRH. So GnRH stimulates the anterior lobe of pituitary to release FSH and LH. FSH stimulates spermatogenesis and also stimulates Sertoli cells to secrete a hormone called inhibin hormone. So this inhibin hormone send the posterior lobe of or hypothalamus to stop secreting, okay, FSH and LH under the influence of GnIH, gonadotropic inhibiting hormone. Whereas the luteinizing hormone stimulates Leydig cells, then Leydig cells secrete testosterone, okay, and this testosterone hormone also responsible for, okay, spermatogenesis and also responsible for development of reproductive organs and development of secondary sexual characters in case of male. So this completes the hormonal control of spermatogenesis. Next, we'll move on to the structure of human sperm. You already know that the human sperms are produced by the process called spermatogenesis. The human sperms are, okay, tadpole-shaped, motile male gametes produced by the process called spermatogenesis. And the male gamete is differentiated into four parts: head, neck, middle piece. Look at the diagram. So it is differentiated into four parts: head, okay, a small region called neck, middle piece, and the longest part of the sperm is tail. The human sperm, which measures about 55 microns in length and consists of head. In the head region, okay, head is oval region. The entire sperm is wrapped by a a plasma membrane, and head consists of two parts. One is a male pronucleus and a cap-like structure called acrosome. acrosome. And this cap-like structure, uh, which is derived from Golgi bodies, which helps in, okay, the penetration of sperms into the womb, because the acrosome releases two hydrolytic enzymes or proteolytic enzyme. One is hyaluronidase enzyme, another one is acrosin. And these enzymes, which dissolves egg membrane and makes microscopic passage. Through this passage, the sperm penetrates into the ovum. So the pale male pronucleus, pear-shaped structure. So it carries genetic information from the father, or it consists of the male pronucleus consists of paternal set of haploid chromosome. So acrosome and anterior of the nucleus is covered by fibrillar sheath. Then head is followed by neck region. So neck is a short part and it consists of two centrioles. One is a proximal centriole and the other one is a distal centriole. So proximal centriole, which helps in amphimixis and also initiates the first cleavage stage. We'll discuss later what is cleavage. I told you in the last session, the diploid zygote enters into the three developmental stages. The one is cleavage, second one is blastula, and third one is gastrula. So cleavage stage is initiated under the influence of proximal centriole. The second centriole, which is present in the neck region, that is called distal centriole. Distal centriole, and this distal centriole which forms the axial filament, and the axial filament which the axial filament which arises from the distal centriole and which forms the central axis of the sperm, or which provides a definite shape to the sperm. Then neck is followed by middle piece, and middle piece mainly consists of mitochondria, and these mitochondria are arranged in a spiral sheath, and that is called nimbecran. Nimbecran. And mitochondria is the only source of energy for motility of the sperms. Motility of the sperms. Is that good? Next one. The middle piece is followed by tail region. So, tail region consists of axial filament which arises from the distal centriole, transverse through it, and helps in the movement of sperms. So, this completes the structure spermatogenesis and the structure of human sperm. In the next session, we'll discuss the oogenesis and difference between spermatogenesis and oogenesis. Thank you.